Hello everyone. I hope you are doing good. In continuation to our discussion about the proteins, today we are going to start the secondary structure of the proteins. Now, before going into the secondary structure of the proteins, it is very important to understand the concept of Ramachandran plot and also the forces which influence the structure of the proteins. Once you have the idea about these two things, then it is easy for me and also easy for you to understand how secondary structures are formed. And then I will introduce you few secondary structures in today's class. All right. So first start with the Ramachandran plot. So if you see Ramachandran plot, it is a graph of torsion or dihedral or rotation angles. So the angles are the same, but they have the three different names and they all denote the same thing. Either we call them as a torsion angle or dihedral angle or rotation angles. Now, what are these angles? If you don't understand, I will explain to you. And this particular plot, it was first devised by the great professor G. N. Ramachandran, right? And now, this Ramachandran plot, it is an inseparable part of protein chemistry. Okay, so it tells about the two angles. One is the phi angle, as I told you in my previous class also. It is the rotation angle around the N-C alpha bond. So here you can see this is the C alpha and this is the nitrogen. So this is the N-C alpha bond, and when the rotation we see or we observe, this is known as the phi angle. And then the psi, which is the rotation angle around the C alpha C bond. So again this is the C alpha and this is the carbon, and this rotation of this particular bond is known as the psi bond. Now, if you ask what is torsion or dihedral or rotation angles, so for those who don't know, they describe the rotation of planes or a group of atoms. Okay, while the bond angle, that describes only the bond between the two atoms, but here they describe the rotation of the planes or a group of atoms. So here you can see we have the two amide planes. One amide plane is this, which is of slightly green color, and another amide plane is this. So we are talking about the rotation of these planes. Okay, now so these phi and psi, they are describing the rotation of the planes or the group of atoms. And you know that the protein backbone is a linked sequence of rigid planar peptide groups. So you can see here this is the main chain, we call it as a protein backbone, and this is a linked sequence of a rigid planar peptide group. So this, the green color planes you can see, they are the rigid, because peptide bonds, they cannot be rotated. Why? As we covered in the previous class, that they have, what is them, they have, yes, they have the double bond character, and because of the double bond character, they cannot be rotated. Therefore they have a fixed plane, and this fixed plane geometry is where all the molecules, they come in the same plane. So here we can see the six molecules which are coming in the same plane. All right, and the another plane, it is again formed by the six molecules, right? So these amide planes, which are called amide planes, these amide planes are fixed, but the angle between the C alpha and N and C alpha and C, they can be rotated. Therefore, they are called as the phi and the psi angles. So phi and psi, they are both defined as 180 degrees in fully extended conformation. Okay, 180 degrees, and they increase clockwise when viewed from the C alpha. Now why clockwise? Because this is the mathematical convention which we are using. Okay, so just clockwise because it is a convention. Now the conformational freedom is sterically constrained. Now sterically constrained means that there are certain combinations of the phi and psi angles that may cause the adjacent residues to either collide or crowd, and when they crowd, they may cause repulsions. So it is basically, the bulky molecules or bulky, groups, when they come in close proximity, then they start repelling each other, or they become, very crowded, so they prevent any chemical reaction. So the proteins has to be a very the conformation which can allow all the molecules to have this minimum collision or this minimum crowding. So this is what the Ramachandran plot tells you. Now what exactly the plot is? It allows to visualize the permitted conformations. So that means which are allowed of the amino acid residues in the protein structure. So this is the actual Ramachandran plot. Okay, and you can see here this goes, so we have the phi and we have the psi, and you can see the values are going between minus 180 degrees to plus 180 degrees. So this is minus, this is plus, this is minus, this is plus. All right, so they can be, why 180 degrees? Because they can be fully extended. So full extension is up to minus to plus 180 degrees. All right. Now only 3 small regions are physically accessible to most amino acid residues. So you can see 3 reasons. One reason is this, one reason is this, and one reason is this. Now you can ask, what is this particular reason? Why this green is at minus 180? So basically, if you see, this is a circular periodicity. Okay. So when you are seeing it in a 2 dimension, but you see that it is a rolled graph, and when it is a rolled graph, then this minus 180 it will touch near 180. So when we increase plus 180 and then we increase 1 degree, then it becomes minus 179, because our full extension is minus 180 to plus 180. So it is somewhere like in the clock, when you see the 1 and 11, they are very near, but if you make it in a linear manner, then they are very far. In the same manner, here 180 plus and 118 minus, they are being looked here as very far, but in actuality they are very near. Therefore this reason is in continuation to this reason. So there are actually 3 reasons: this one big reason, and then one intermediate reason, and one the small reason here. Clear? Good. Now the blue shaded regions, so you can see here, this is the blue shaded region. These are sterically allowed phi and psi angles for all residues except the glycine and proline. So these are the 2 exceptions. Why they are the 2 exceptions? I am going to cover them shortly. Now the green shaded regions, here you can see this is the green shaded region. They are more crowded because that shows the outer limit of the phi and psi angles. So most of the proteins, they fall within this particular regions here, right? And the yellow one, this is the alpha, that represents the right hand alpha helix. So this is the right hand alpha helix region. So most of the proteins, almost all the proteins right handed alpha helix, it will fall within this particular phi and psi range. Then alpha L, this is alpha L, this is the left hand alpha helix. So this represents the left hand helix. Then here we have a small c, which is a collagen helix. Then the same arrows, same directions, they are the parallel beta sheets, and these are the anti-parallel beta sheets. So these are some of the secondary motifs which are representing here on the Ramachandran plot. Okay. Now there are some notable exceptions. The phi angles are around minus 60 degree for proline. So we are talking about this particular angle, phi angle, and minus 60. So minus 60 will fall somewhere around here. Okay. So for due to the cyclic side chain, and the glycine is the only residue without the C beta atom. Now why it is the only residue without C beta atom? Simply because it has hydrogen as a side chain. It has no carbon or carbon group there. Therefore there is only one alpha carbon, there is no beta carbon here. So that is the only residue without a C beta atom. And the permissible range of phi and psi, it covers a larger area. Okay, it covers a larger area because it is the simplest, and it gives the flexibility. So polypeptide chains at glycine, they assume conformations which are forbidden to others. So that plays very smart role in the protein structure. So what other amino acids cannot do, this is small amino acid can do, and therefore it is very, very useful. Okay, I hope I have made this clear to you about the Ramachandran plot. Now let us understand what are the forces which influence the protein structure. So there are several different non-covalent interactions. We are talking about the non-covalent interactions which stabilize the protein structure. They are hydrogen bonds, hydrophobic interactions, electrostatic interactions, and van der Waals interactions. Now let us understand, how they are important for the protein structure. So a hydrogen bond is basically a covalently attached hydrogen atom to a highly electronegative atom, and these electronegative atoms are O and F. So you can just abbreviate them, or for mnemonics you can just learn them like FON, phone. They are almost, a hydrogen atom will be attached to these either oxygen or nitrogen or fluorine. Okay, and then it becomes partially positive. So you can see here, this can be A can be O, N or F, and it is attached to the hydrogen atom. So once it is attached to the hydrogen atom, this hydrogen atoms become partially positive. So you can see here the partially delta positive charge here, and they are, because electronegative, so they will they will attract the electrons from this hydrogen. So that will create a negative charge on this A, which is O, N and F. So you can see here delta minus. This is clear here. Okay, now once it has developed a partial positive charge, this hydrogen atom then attracted to a lone pair of electrons on another electronegative atom nearby. So here you can see this is again a electronegative atom, and it has a lone pair of electrons. Now because it has a partial positive charge, so it will be attracted to it, and it will form a hydrogen bond, and that's how the hydrogen bonds are formed. Okay, so they are generally made wherever possible within a given protein structure, wherever possible. Now if you see the secondary structure, they are stabilized only by the main chain to main chain interpeptide NHCO hydrogen bonds. Remember, in case of the secondary structure, it is only the main chain or the backbone. So backbone to backbone interpeptide NHOC hydrogen bonds. So you can see here, this is the main chain, and here you can see this is NH of one residue, this is the CO of another residue, and they are forming a hydrogen bond. In the similar way, here they are forming the hydrogen bond, but that is happening only by main chain to main chain interpeptide bonds here. But when we talk about the tertiary and quaternary structures, which are a higher level structures, then they are held together by the hydrogen bonds of the type main chain to main chain to side chain, and side chain to side chain. So they can form almost in any configuration. But for secondary structure, it is only by the main chain to main chain. Okay, so this was about the hydrogen bonds. Now, if you see the hydrophobic interactions, they cause hydrophobic moieties to aggregate or cluster. So we have the non-polar amino acids. Okay, and these non-polar amino acids, they can be aggregated by this hydrophobic interaction. So by this aggregation or clustering, they are minimizing the contact of non-polar residues with water, and therefore this interaction is highly favorable. Okay, now you can see here, this is the hydrophobic side chains, and these green are the hydrophilic side chains. So once they come in contact with water, then all these hydrophobic side chains, they cluster together, so that they can be minimizing the interaction with the water molecules here. Okay, so this is known as the hydrophobic interaction, and this is very important because the side chains of these amino acids, they are usually found in the interior or core of the protein structure, and they are almost exclusively hydrophobic. Okay, and the protein surface, you can see here, this is the protein surface. It may consist of both polar and non-polar residues. All right, polar and non-polar residues. So therefore, these hydrophobic interactions, they are very important in stabilizing the protein structure. Okay. Now, the third is the electrostatic interactions. So they are the attractive or repulsive forces between the charged particles because they are electrostatic interaction. So therefore electro is involved here. So we are talking about the charged particles here. So you can see here these are the positively charged, they are the negatively charged, and we have the charged amino acids. Well, when they are in solution, they are the basic amino acid side chains, for example, lysine, arginine, and histidine. Then we have the acidic side chains, like in case of aspartate and glutamate, they become aspartic acid and glutamic acid, and the amino terminal and the C terminal of any peptide. So they may exist in ionized states and they can carry positive or negative charges. Okay, they can carry positive or negative charges. Now, non-polar residues, so what we understood in the last slide, they mainly stabilize protein folding by hydrophobic interactions. But when we talk about these electrostatic interactions, these charged residues, they provide specificity. Remember, charged residues provide specificity. And with polar groups, they often participate in the hydrogen bonding. Okay, so they can act over relatively long distance, so they can guide molecules toward each other, right, in the solution. They can increase binding efficiency, and they can affect the association and the dissociation rate constants in any enzymatic reactions. So they are very, very useful in enzyme substrate interactions. Okay. They are very useful in protein protein interactions or protein protein binding, and they are very useful in antibody antigen recognition. So electrostatic interactions, they play the role to provide the specificity, while the non-polar residues or the hydrophobic residues, they stabilize the protein folding by hydrophobic interaction. So these are the two very important points as a takeaway you should carry with you. And other aspect is the DNA binding proteins. So when DNA binds to proteins, usually that happens, in molecular biology, you must have heard about the transcription, or where the DNA is binding to the protein, or when the DNA is coiled, right? So when in the histone proteins or some non-histone proteins, transcription factors, they all bind to the DNA. So that is also important because of this electrostatic interaction. Right. Now, the last one is the van der Waals interaction, and we call them as the London dispersion forces. Okay, you can just call either van der Waals, or you can say them like London dispersion forces. They are also the electrostatic forces, but they are caused by temporarily fluctuating the dipole moment. Temporarily fluctuating the dipole moment. So a brief shift of orbital electrons to one side, it creates a similar shift in the adjacent atom. So you can see here, this is atom 1, this is atom 2, and here the atoms are polarized. So you can see a brief shift of electron in first atom, it is also creating the similar shift in the second atom, and when that happens, then there is a electrostatic attraction which is very, very weak, and it is temporarily, but this is known as the van der Waals interaction. So this is a very non-specific interaction. It is not a very specific interaction. It can form between any kind of molecules regardless of the chemical structure. Whatever the chemical structure is, doesn't matter. Van der Waals interaction can be formed. And particularly in proteins and biomolecules, the close packing of hydrophobic side chains are assisted by this van der Waals interaction. So basically, because hydrophobic side chains are found in the core of the protein, and when they are stabilizing the three-dimensional structure of the protein, which we will see, later. So this close packing is again, facilitated by this van der Waals interaction. Then they also stabilize the protein interior, and in molecular recognition, where many weak contacts, they act together. And why it is important? Because if the weak interaction is there and few weak interactions are there, then probably it is not specific. But when there are so many weak interactions, they stabilize the whole interaction, and because of that, the proteins they can act or they can activate, okay, or inhibit. So that happens because of these weak interactions. So in general, these forces get stronger as the number of electrons in a molecule increase. Now, why it happens? Because when there is a number of electrons in molecules increase, so the total number of electrons are increased, and that also increase the relative molecular mass, and that because of this the strength of the van der Waals forces are increased. Okay, so all these four forces, the hydrogen bonds, hydrophobic interactions, electrostatic interactions, and van der Waals interactions, they are very, very weak. They are non covalent. But in actual molecular biology and biochemistry, they are the most important forces which are involved in not only stabilizing the structure of the biomolecules but also to carry out the metabolic reactions here. Okay. Okay. Now, once you understood the Ramachandran plot and the forces, now I will introduce you to the secondary structure of the proteins. So, what is the secondary structure of proteins? It is basically a common regular local folding patterns or repeat structures of the polypeptides. Remember, common regular local folding patterns. Okay, they are very regular or repeat structures in the polypeptides. So, a regular secondary structure occurs when each dihedral angle phi and psi they remains the same or nearly the same throughout the segment. Remember, in each of the regular folding, the phi and psi they remain the same or nearly the same throughout that particular segment. Okay, then only the regular structure happens. So only few types of secondary structures are particularly stable and occur widely in proteins. They are most commonly the alpha helix and beta pleated sheets, and these two were first proposed by Linus Pauling and Robert Corey. Okay, so alpha helix and beta pleated sheets they are the most common secondary structures. So you can see here this is the helical structure and this is the pleated sheet structure. Okay. Now some structures like beta turns or omega loop they are also identified, but they contribute with alpha helices and beta sheets to form the final protein structure. So this is one example of the omega loop here. You can see here there is a looping, okay, but they are helpful in the final structures of the protein. And if there is no regular pattern, then the secondary structure is undefined or a random coil. So this is something a random coil structure or undefined structure. So these are the secondary structures which we are going to take in the next class. Okay, and in details we will understand how these structures are formed and what are the features of these structures, right? So in case if you have something to ask or something to understand, again you can see this video, otherwise you can write to me and I will try to clear your doubt. So I'll see you in the next class. Till then, take care. Bye bye.